I don't think I have to introduce Dana Peer to many of you. I will still do the honor, because I did my homework. I looked up your long CV, but I learned that you did actually a bachelor in math and doing a master and PhD in computer science, Dana. That was still in Hebrew University in Jerusalem before.
My ancient history before I forgot all my math and became a biologist.
Well, I knew this, but I kind of forgot that your PhD advisor was Neil Friedman. The guy's a legend, obviously, right? He's a pretty impressive guy. And then she did all kinds of fantastic research stays with church, working at Harvard Med School, but then at some point making lab in Columbia, then Memorial Sloan Kettering, and has been one of the, I think, leading figures for single cell analysis in the field. Even in early times already, I don't know if you guys remember that systems biology times, working on fax data, trying to learn something about interactions, potential causality, trying to do those models. And I remember her, I think, seeing one of the first times, a keynote lecture. I think it was at one of the ISMBs or so. Like all those things so nicely slotting together in her CV and building upon each other, and then obviously those beautiful pseudo-time algorithms. Basically, in pretty much every one of my talks, I say, we usually name our algorithms something like diffusion, pseudo-time something, and then she, non-native German speaker, comes up with this beautiful term of Wanderlust. How cool is that, right? Wishbone and so on.
That was actually my grad student and not me. All credit to Elad.
Okay, you have good grad students. In any case, there's been a lot of obviously wonderful work coming from Dana's lab, but I think I should also super briefly mention organizer quality, in particular strong contributions to the analysis partner, Human Cell Atlas, and so on. So we're really excited to have you here and looking forward to talk about trajectories and gene regulation. Thanks, Dana, for taking the time.
Yeah, Fabian, you really set me up. First, you put me right after two young speakers who really put in a lot of efforts into making perfect, polished talks. So, my talk is going to be less good. And then, with that introduction, I can only disappoint. So I actually do, you introduced some of the early work, and, there's a lot of talk about, pseudo-time is old school, is dead, it's not worth it. So I actually do want to give a little bit of a plug for pseudo-time before we go into the more modern things. And this is really the reason that pseudo-time works. The first time we noticed this is, again, with CyTOF data. This is only 13 dimension. And we really, noticed that one of the amazing things in single cell data is that we have a continuum of cell types. The cells taken out of a tissue are asynchronous, and that gives us this amazing power. And the developmental signal is so strong that even if you use PCA, you get something, in this case, the B cells, even along PCA, seem to follow something that resembled a developmental trajectory. Of course, PCA isn't good because the development and the space is very, very non-linear in its nature. That's why we switched from PCA to diffusion components, which, Fabian really brought to the world. And in order to get these diffusion components, of course, I'm trying to go through some basics, ancient history really fast, just to make sure everyone's on the same page. So because of the nonlinearity of this space, we really switched to neighbor graphs and to diffusion components and sort of directions of variation along these graphs. And we traverse them with paths to look at differentiation and learn global structure on this sort of manifold that's really, really, really well captured by the data because of the asynchrony and the continuum. And this is really powerful. So, this is actually Wanderlust, which I'm showing you, where, you can really reconstruct very accurate progressions in order. You can't get real time. You can only get order. The timing, this is before that, and, velocity can actually give you some timing. But again, if you have a lot of cells and you have an accurate algorithm, you can get something, quite accurate. And one of the reasons I like this work, and I like highlighting it, even though it's ancient history, is that we've managed through this accurate timing to get a really, really tiny population, about 7 in 10,000 cells. Again, this was enabled by CyTOF and shows why we need so much. And in this tiny population, a lot of important stuff is happening. The VDJ is recombining, the DNA of the B cells are recombining, and there is a completely different signaling checkpoint. And the signaling checkpoint only happens in these cells and no other cells. And this is an important checkpoint that goes awry later in pediatric leukemia. And just to show you just how rare this population is, this is sort of all the cells we collected. This tiny, tiny red slice right here is the progenitor B cells, and the population where this action is happening, where the signaling checkpoint is happening, where the VDJ is recombining is this tiny little pie slice of this tiny, even tinier pie slice, going to show why we need so many cells and why we need a project like the Human Cell Atlas, which will collect about 10 billion cells, and why we need really good and accurate algorithms to recover it. Again, Pseudo-time is so wonderful that we can get dynamics. This is just throwing TGF beta on a cell line, and in day 8, getting the entire continuum of epithelial to mesenchymal cells, everything in between, the de-differentiation, the stem cells, and all the behavior of all the regulators and transcription factors and chromatin modifiers. And we're getting dynamics from a single time point here because of this asynchrony, and this dynamics is so strong that even though this pseudo-time actually isn't that accurate, I can tell you, we're still able to recover gene regulation. We're able to predict, combining the covariant structure of the data and the sort of pseudo-time to get sort of temporal causality. We're able to get, you know, significant overlap with independent ATAC-seq data for most of the transcription factors. We got a pretty good prediction of their gene targets just from this one time point in a cell line using pseudo-time, just to show the power. Now, going a little further, I sort of want to, again, give the concept of Palantir. This is work by Manu Sethi, who's been a wonderful postdoc in my lab. He's about to start faculty in the Hutch, and I highly recommend his lab for anyone who wants a young and energetic advisor. And I want to, you know, say a couple, you know, stories about him. So this is actually a little bit similar to the CellRank, where we really want to use a Markov chain to understand potential future cell fates. Like, our goal here is to take any cell in our data and try and figure out, you know, where it will go, what its future potential fate will be. And, you know, we've built neighbor graphs, and we've built pseudo-time along these neighbor graphs, and a Markov chain needs a directionality. So here we simply use pseudo-time for our directionality. And at the end of the day, with all its problems, actually for development, it works quite well. It has two really strong assumptions. One, that we sort of know the starting point, and two, that the cells don't de-differentiate. And these points are broken in regeneration and cancer, but actually for the study of development, I find these approaches very powerful. And just to show you the sort of Markov chain and the powering of the network that sort of Volker showed you with such nice math, I actually find it very intuitive to just see math very visually. So here we have all the cells and all their future potential cell states based on one step in the Markov chain, based on going, you know, one step away in their neighbor, based on their probability to go in each step. And as we power this network, as Volker said, in brute force, but very visually, you can see how the cells can go farther and farther away, and there's a lot of stochasticity, there's a lot of plasticity, there's a lot of uncertainty towards future cell fate, but we can actually get, you know, for each cell, when this thing converges, what is its sort of future branch probability? And there was a question earlier, like, how much can you trust it? And I actually trust it a lot, because at the end of the day, the cell manifold that captures the dynamics of the cell is really powerful. We have typically almost all intermediate steps, certainly during development and regeneration, in a particular data set. And while we don't have the epigenetic layer, the way I view it is, yes, there might be a few paths to a particular cell state, but if we treat it as a cell state, irregardless of how it got there, and saying, given that I'm in this transcriptional state, what is my probability to go to each direction? This probability is pretty robust, and again, it's probabilistic. Cells are stochastic, and we've actually tested this approach quite extensively across cell tagging, and the probabilities that were predicted by Palantir, and I don't have the slide here, but I'm just relating to a question from the previous talk, actually beautifully matched the probabilities by the lineage traced cell tagging method by Samantha Morris. And so at the end of the day, what this gives us for each cell, and this is why I really like this approach, is we know what is its probability to reach each terminal fate. Of course, if it's very high up, if it's very progenitor, right up here, this is a cartoon of hematopoiesis, then, we don't know where it's going to go. It can go into any one of its terminal hematopoietic states, and there's a lot of entropy and uncertainty. And as you go down, the fate becomes, going in one direction or another. But when we study the data, even something like hematopoiesis, we see that actually there's quite a bit of uncertainty and plasticity, even quite late in the game. And cells are more plastic than typically modeled when we look at a lot of systems. So I like developing methods and applying them to real biology. That's why I moved to Sloan Kettering. I mean, yes, I have an undergrad in math and stuff, but the real joy is when you discover something new. And one of my favorite systems in the world is embryogenesis. And the gut tube is the coolest system in the world because it's about 3% of your cells. And again, this sort of happens in early gestation. And you see this sort of purple outline right here at embryonic day 7.5. There's this cool thing where it inverts inside out, and this external layer suddenly becomes an external gut tube. It just like, boom, switches in. And then you have this tube that's about 3% of your cells a few days into development. This is, of course, mouse development. And out of this tube, bud and branch all your major internal organs, your thymus, your lung, your liver, your pancreas, your intestine. And suddenly, basically, you have organs budding out of this endoderm. And so we tried to look and understand this development and how cell fate decisions are made, how cells decide to become endoderm, how these endodermal cells pattern themselves and eventually form all these major internal organs. And, we used Palantir to look at some of the earliest stages. This is still an ICM blast. People viewed it as something very, very homogeneous. And when we looked at it, we found that actually it's not homogeneous. And even pretty early along, even at the very, very earliest time point, cells sort of already had a propensity to go to either embryonic or extra embryonic fates. And again, the nice thing of having a pseudo-time, a nice thing of having an ordering of the cells, if this ordering is accurate, is you can actually look at, how do regulators change along the pseudo-time? And you could try and understand not only what's going on and where cells are going, but what are the regulators and what are the regulatory events that have to happen in order for this to happen. And here we're looking at how cells become primitive endoderm. And, people knew that the FGR receptors were very much related to it. And this dotted line is like the cell fate probability to become a primitive endoderm. And what we see is that the cell becomes a primitive endoderm when it combinatorially, and combinatorics is critical for development, expresses both FGR1 and FGFR2 simultaneously at the same time. And that's sort of when that lineage decision is made. And in follow-up decisions, we saw that indeed, in order to lose this particular lineage, you really need to sort of combinatorially knock them down, and then you really lost any ability to create this cell fate. Going further down and taking another day, these are the extra-embryonic and embryonic epiblasts are really different. And, if you look at the yellow cells, which is day 4.5, they really diverge. But one of the things that we found is this bridge right up here. And, we thought it was a possible transdifferentiation. Our biological colleagues, who are amazing, Sonia and Kat, thought that no, this is crazy, this is noise. There's no way that cells that are sort of epiblasts, embryonic stem cells, suddenly become cells that will later become placenta or something. But our pseudo-time said that there was a path from embryonic to extra-embryonic cells, and in fact, somewhere here at this point right here, there seems to be again some plasticity, some differentiation potential to choose different fates. And again, the branch probability said that epiblast turns into extra-embryonic visceral endoderm. And so we finally convinced them, and then they did lineage tracing experiments with 2 systems and multiple replicates. And we consistently saw that epiblast cells sort of somehow traveled to this surface, to this sort of external membrane, and completely switched their fate to visceral endoderm. They had the epiblast, lineage tracing marker, but then they started expressing all the visceral endoderm key markers and regulators, and started also morphologically behaving like visceral endoderm. So going forward, this is great, and I just tried to show you how wonderful, even simple pseudo-time is, and how much biology you can get out of it. But it's broken in regeneration and cancer, which has become something that I'm studying extensively. And we need to know a starting cell, which we don't always know. So this, here comes velocity, and we just heard two amazing talks and about velocity, and it's, I think, one of the most important discoveries in computational biology, and certainly the past 5 years, and competitor of the decade. And one of the problems with velocity is that you get a single velocity, but it's not constrained to the manifold. And I just showed you about 10 minutes of just how much you can learn from being constrained to the manifold. And not all velocities are within the cell manifold. Not all the velocities are even consistent. There's some areas like right here where velocities are all over the place. Neighboring cells seem to not agree where they're going and not remaining in the manifold. So this is why CellRank, which we just heard before, and I'm going to give it again because right now CellRank is my favorite algorithm, period, exclamation point. And this is work with Marius and a wonderful fun collaboration with Fabian, our host. So, I luckily don't have to explain all the math, but again, this comes in context of Palantir. And here, what we do is instead of using pseudo-time, we actually use velocity. We actually use something with real dynamic information to try and orient where we're going. And this really gives us these velocities that really propagate over the manifold. And to answer how do we know that what's going on with a single cell, this manifold and the ability to really have all the synchrony and all the intermediate cell types and propagate behavior on the population of cells and the distribution of this population of cells, it really gives us a lot of robustness, really allows us to really overcome a lot of uncertainty and really infer future fates along this manifold. And again, using the global structure of the manifold and embedding our velocities within that and propagating our velocities within this global structure really allow us to overcome areas where I just showed you earlier, where velocity is uncertain and messy and not that great. And again, in CellRank, we can model this uncertainty and say, this region is very variable. This region, none of the neighbors agree where they're going. So let's actually, put this velocity directly in our algorithm and be a little bit more diffuse in terms of how we model the directionality and let the probability be a little bit flatter. And again, because we're propagating this in the context of a Markov chain along a global structure, which many regions is very certain, and it has to have some type of global consistency. This improves our predictions and really help us overcome these sort of noisy, uncertain areas. So again, what can you do in this? I think this is really powerful. And Volker sort of really quickly went over this. I'm going to give you another example. We want to know, how you get to these purple cells. And when you look at these velocity vectors and try and figure out how to get to the purple cells, well, it's really hard to figure that out. But again, if you have these fate probabilities, you can actually see the path in the data. And if you look at, what genes, what regulators change along this path, you can really capture the known sort of master regulator of delta probability, showing just how powerful this is. And again, the ability to look at the dynamics of these different regulators as cells go through one probability or another. And these are sort of plots plotted based on the probability, the cell fate probability to go each of these fates, really captures the master regulator for each one of them. Now, I told you guys that, the real reason we want to do CellRank is for regeneration, because that's where pseudo-time is very unreliable. And so, one of the things that made me very happy is this result on lung regeneration. And and this was a collaboration with Herbert Taylor, where we saw that actually a goblet cell was de-differentiating to a basal cell. And this is the opposite direction of differentiation. This is a clear case where a much more progenitor earlier stage is actually the velocity is pointing the opposite direction, which, Pseudo-time would miserably fail here. And, Herbert fortunately, did some imaging to really show that these transitional populations, which we predicted with all the regulators, really do exist in the data, showing the importance of being able to actually model time not through pseudo-time and not make such strong assumptions in regeneration. Now, I really love cancer, and this is a take home, which I've said 3 times. So, we've been applying CellRank to cancer, and we have a few cases where it succeeds, but I really want to quickly give you a case where it miserably fails. And this is actually lineage plasticity, where lung cancer cells become dedifferentiated and switch complete fates, do a complete lineage transformation, and become resistant. And so we wanted to study this in an organoid model. And what I wanted to show you here, and I'm seeing I'm running out of time, so I'm running through this to get to the final thing, is that it's that cells in cancer become a lot more plastic. You saw very clear paths in sort of these healthy cells, but if you look at the neighbor graphs, once you have cancer, all plasticity is lost and cells can go all over the place. And that's why we really need good methods to orient directionality, because all bets are off in directionality. And I'm going to skip this. We could predict what pathway matters and fix it. But really, in this particular case, again, CellRank failed, and I was very, very miserable about that. So we asked, why does CellRank fail? And this is, I think, Yuri told it very nicely. It matters which cells are driving it. Not all cells have strong splicing signal. Not all cells are driving the velocity vector. And in hematopoiesis, it's this one case where pseudo-time works beautifully and velocity consistently and miserably fails. And it's not fails because it's uncertain, it fails because it's certain, and it's certainly going in the wrong way. So I like looking at the data and understanding what's going on. So here you really see the rise of a cascade of transcription factors that are driving the cells towards erythroid fate and their targets. And you can really see, again, this dynamic rewiring of the regulatory network. Here we have the covariance matrix of each regulator and its targets. And you see that over time, in each time window, we see a completely different covariance structure. We see a completely different regulatory network. And this regulatory network is really driven by rewiring of the epigenetic landscape. This is ATAC-seq data over the thing. And you see really that these peaks open only in late stages. The ability to even regulate and turn this gene on only happens in late phases. Here, in the regulator, you have a little bit more, you have some poise promoters, you have some ability to map early, but then you have this very, very specific peak for the erythroid in the ATAC-seq data. So we have dynamic regulation, we have ATAC-seq that's driving this dynamic regulation. So this is, I'm going to run through attack velocity really fast. So I'm like, okay, can we get these vectors from ATAC-seq? Can we use gene regulation and the idea that transcription factors activate their targets? So if we know the transcription factors and the regulatory network, we can predict where cell fate will go. We're going to model it as a linear equation. It's not perfect, but it's robust. It has a closed form solution. And now all we need to do is figure out this A matrix, which is sort of the regulatory dynamics of the system. If we figure out this A matrix, then knowing the transcription factors and the potential regulatory network of a cell, we can know where it's going in the future based on its regulation. So how can we figure out A? Well, we need to figure out its structure. And this we can get from the ATAC-seq data and the motifs in the open chromatin, really limiting and giving a prior on which regulators can regulate which gene. And we need to know the magnitudes. Here we have 2 unknowns. What are the slopes? What are the impact? And what is the regulatory network itself? Now, of course, when you have 2 unknowns that are related, the most wonderful thing is is EM. It's a very powerful method when you have too many latent variables that are interconnected. Key to success is a good starting point and good constraints. And we don't have this algorithm fully down. This is work in progress. This is a workshop. But really, on hematopoiesis, it works really, really well, both globally and at the level of individual genes. And so right now we're really driving and working on using regulation as a vector to understand the future, because, you know, it's a very powerful signal. Genes, transcription factors, and regulation actually determine future states. So we can try and use that itself directly, and the signals that we have within, combining epigenetics and the observed covariance structure between genes to try and figure out the regulatory network and use it to orient the dynamics in future cell fates and their probabilities. So this is a quick summary. I'm going to skip that and really put up the thanks. You know, this was, I presented the work from Manu, Cassandra, and Joe. Wonderful collaboration with Fabian and Marius, and thank you, everyone.
Excellent, thanks, Dana. This is such a bold move to go from those univariate single gene models that we've been playing around, let's say a little bit coupling with the latent time, but now really going towards adding regulation. I mean, I think it's like has been my old dream since was really nice. There have been a few questions people just put in the chat, but I think Nikolaus Rieski has first question. Nikolaus, can you go ahead? We need to unmute you.
Okay, so maybe you can hear me. I don't know.
Yeah, I can hear you.
Okay, great. Hi, Dana. Super nice talk. Thank you so much. And I just wanted to ask, so you know, I think that what you're doing that is so important. Coupling expression to explain the data really, and in the end coupling it to sequence. But the model you wrote down is you're assuming somehow a linear interaction between the transcription factors, it seems to me, if I understood that correctly. But that is, I mean, you said it yourself, for example, development is all combinatorics, and you know, it's highly non-linear and so on. So how do you see that? So
it's all a question of resolution and what your question here is. And here I'm really using, I'm not saying I'm going to, you know, perfectly predict the right regulatory network, but I'm using regulation to try and actually, as a vector field, to try and orient and get the directionality. Now, I've done a lot of tests, and I think I'm not the only one that have shown that while we have non-linearity in regulation, often because the regulatory model is, the linear model is so robust, then often for non-linear systems, the robustness of a linear model, when you learn it and you fit it to data, is closer to the ground truth than the nonlinear model, again, because you have to fit this to data that you originally gave the system. That's one. Of course, the linear model has a closed form. And as a global first approximation, just to see where things are going, I believe that this is good enough. If I want,
I mean, you have to be honest, right? At the moment, it's an identity matrix that we use, so we completely decouple. So I guess,
yeah. So of course, when we want to zoom in and really model regulation and what's really going on, and when I want to really capture the regulators and not just the global structures and future cell fates, then of course we have to go through to more sophisticated models, where here the goal is not to get a global map of cell fate and velocities, but here the goal would be to actually get correct regulation. And there I would use different models, and there I would use models that allow for combinatorics, certainly in the context of development, but that's a different goal and a different model.
There's a whole bunch of questions, maybe short question, short answer before we proceed to the coffee break. Lale Hakverdi has the next question.
Yeah, hi Dana, thanks for the nice talk. So just to maybe get it once more. So is that correct that you think that the fact that in your example for the purple population we don't get the RNA velocity with RNA velocity is good direction? Is that because we with the RNA velocity we are kind of getting instantaneous velocities, which is on the time scale of a splicing time scales, and then you think with the ataxic data you can get velocity on average velocity in a longer time? Is that the essence?
So actually, no. So in the case of hematopoiesis, and I think, Georgia also sort of mentioned it, velocity really depends on what genes are spliced. And, the genes that have a lot of splicing reads, where your velocity signal is coming from depends on like how much exons and how many spliced reads you're able to get. And again, if you imagine these diffusion components, in sort of neurodevelopment, you have splicing up the wazoo. That's why it worked so beautiful in the neurodevelopment context. But if you can imagine, again, with diffusion maps, imagine you have a diffusion component and all the genes that really change along this diffusion component don't have any spliced genes. And so you have no way to capture the directionality of this diffusion component correctly. That will mess up your entire velocity field. So what I think is going on is some biological systems, their key genes are better captured by splicing reads than others. And if you don't have the splicing reads on the genes that you need to characterize what direction the system is going along each critical diffusion component, then you're a bit screwed. And in the case of ATAC-seq, again, it goes back to be genome-wide. It's all a matter of, one of the advantages of Pseudo-time, for example, is again, it's genome-wide, where in velocity, your signal is more limited to a smaller subset of genes, which you can hope are the relevant ones. And when they are the relevant one, it's the most beautiful thing in the world. And when not, then we're going to have to go other directions.
Thank you very much.
There was a short question from Sanchit Misra about computation intensity. How much compute time do you need? Would you sort of profit from faster machines?
Not for any of these algorithms. I have a lot of algorithms where more compute time would be wonderful, but these are actually pretty efficient, all of them.
There's maybe that's sort of reaching coffee break. Just a short last one from Geneviève Stein O'Brien. Geneviève?
Yeah, I was just wondering, so in terms of directionality within ATAC, that's really a very interesting question, but is it possible to link the directionality from the ATAC to the expression? Have you looked at that at all in terms of, whether the epigenetic markers are regulating the expression and vice versa, and whether there's any sort of delay in timing in those effects?
So we haven't collected enough data to sort of really link that. I mean, the single cell ATAC-seq data is a lot newer, so we're still, figuring it out. And it's definitely a lot more complex, particularly because we have extensive poised promoters. So generally speaking, understanding ATAC-seq data is something my lab is very engaged in, and it's not as simple as you would think before we entered. For the context of attack velocity, we're really using the attack very weakly as just sort of limiting the potential regulators of each gene. It's actually very weak, and most of the signal under this constraint is actually driven by the covariance matrix and not the attack peaks. But we are working on methods to really use the attack in a much more powerful way. Step number one, understand it and make sure that the assumptions you're making on it are true, which have been broken in my case. Well, so we're still figuring out the best way to model this.
Thanks, Dana, for a wonderful talk, and thanks to all the speakers in this session.